Good afternoon. My name is Aaron Osborne. I'm the Chief Medical Officer at Adverum Biotechnologies. We're a public gene therapy company that is located in Menlo Park, just down the road from here. This presentation will contain some forward-looking statements. I'm going to spend most of the presentation talking to you about ADVM-022, which is our intravitreal gene therapy for VEGF-driven ocular diseases. But at Adverum, we're pioneering novel gene therapies for multiple diseases, mainly ocular. We also, besides having ADVM-022 in wet AMD, and I'm going to be presenting some data in a moment, and we also tomorrow here at the AAO Subspecialty Day in Retina, will be presenting additional 24-week data, and that's something that very much looking forward to. I also want to talk a little bit about the industry-leading platform that we believe that we have and our capabilities. Specifically, we have an AAV7M8 vector which features within ADVM-022, and that has been developed using directed evolution, both within, but mainly within Adverum Biotechnologies. We have a robust patent portfolio, and we have a scalable manufacturing process. And besides being a science-driven company, we have a leadership team who've got an extensive ophthalmology experience and drug development experience. We're well capitalized with $177 million in cash. Let me set up the problem. And the problem with anti-VEGF therapies is not that they're ineffective. They're incredibly effective. And in fact, they've reduced global blindness due to AMD significantly. But what we're seeing increasingly is that the real-world outcomes don't match up to those in the clinical trials. In this graph here, you can see the great double-digit gains that we see with Lucentis in the pivotal trials in Anchor, Marina, and Harbor. But when the frequency drops down, those gains are far less robust. And that's not just in the clinical trials. What we're seeing increasingly in the real world is that it cannot be delivered at the injection frequency required to maintain vision. So what's actually happening is that the gains are less in the real world, and then patients are beginning to lose vision, often going well below baseline as they progress their treatment. And wet AMD is a lifelong disease. I think that's a really important point here. The aim is to preserve vision until patients reach the end of their lives. So we really need a solution that can reduce the treatment burden and potentially allow patients to maintain their vision much better than they do with existing therapies. And that is why ocular gene therapy is something that's very attractive and being looked into. At Adverum, we believe the intravitreal approach offers significant advantages over subretinal, specifically in that it can be delivered to a much broader patient population, and there are some safety advantages as well. And here in the situation where VEGF is a secreted protein, and we know that anti-VEGF, when delivered intravitreally, can treat the disease effectively, we believe specifically here intravitreal offers significant advantages. And here is the technology. So, as I mentioned, ADVM-022 was developed using directed evolution initially at UC Berkeley and then continued at Adverum. And it's been developed specifically to enable efficient intravitreal delivery, to increase the transduction of retinal cells, and to increase the protein expression. And here the protein that's being expressed is the aflibercept protein. We have a very robust preclinical data package, and I'm going to share a couple of highlights here. As you can see here on the top line, there are two dotted lines, and those represent aflibercept concentrations from a bolus of Eylea that was delivered to a non-human primate. Those dotted lines represent therapeutic levels, and they are at approximately 4 to 5 weeks after that aflibercept was given. These are vitreous concentrations of protein. The solid lines represent the protein that was measured following an ADVM-022 injection. And this goes right the way through to 30 months. So you can see here that there's a consistent and sustained protein expression right the way through 30 months. And besides showing levels that are thought to be efficacious, we've also seen efficacy. So on the right-hand side, you can see that ADVM-022 here was delivered 13 months prior to laser being administered in an NHP laser CNV model. And the efficacy seen with that was comparable to Eylea when given at the time of lesioning, which is 1 to 2 weeks prior. So here we're seeing sustained expression, and we're also seeing efficacy in the laser CNV model. Let's fast forward to humans in the OPTIC trial. So the OPTIC trial is an ongoing phase 1, first-in-human trial looking at ADVM-022 in patients who require frequent intravitreal injections to maintain their vision. We've completed Cohort 2 enrollment, and we look forward to making announcements about subsequent cohorts here at AAO tomorrow. We presented the 24-week data at the recent Retina Society conference, and again, we'll present additional outcomes tomorrow here at AAO. Of note here, patients are given a prophylactic oral corticosteroid course at the start, and from month 1, they are eligible for rescue with aflibercept bolus. These are the baseline characteristics, and really the key takeaway here is that these were patients who were requiring a number of intravitreal injections to maintain their vision. In fact, that's an average of 6.2 injections over the 8 months prior to screening, which translates to an annualized average of around 9.3. They had relatively good vision, which I think is important as well. This is better vision than you would see, for example, in treatment-naive studies such as Hawk and Harrier, because these patients have already been treated, they've reached a plateau of vision, and really the aim for these patients is to maintain that vision. We would not expect to see vision gains. The central subfield thickness was slightly elevated compared to normal. Some of these patients had exudation at baseline, despite receiving frequent injections. In terms of safety, obviously the most important outcome for phase 1 studies, ADVM-022 was safe and well tolerated. With regards to systemic adverse events, there were no drug-related non-ocular adverse events and no serious adverse events. Looking at the eye, although inflammation was commonly reported, it was generally mild. It also responded and was considered manageable by topical steroids, and there were no severe AEs. There were no signs of retinitis or chorioretinitis or choroiditis. When we look at the vision, so the aim was to maintain the vision, and through 6 months, that visual acuity has on average been maintained with a point estimate of -2 and the 90% confidence intervals crossing 0. In terms of CST, there were actually improvements of -50 microns. I'm just going to present a couple of cases here from this first cohort. This was the Sentinel subject, the first patient treated with ADVM-022. We have a retrospective study which shows the treatment and outcomes on OCT prior to OPTIC. And as you can see here, there's a pigment epithelial detachment, there's subretinal fluid, despite treatment every 5 to 6 weeks with anti-VEGF. Once they come into OPTIC, they are treated with 022, and as you can see, the subretinal fluid starts to subside. By week 12, that fluid is resolved, and it remains fully resolved through week 24 with no rescue injections administered. Another patient that I want to show you is the patient that had the most injections prior to coming into OPTIC. This patient really illustrates the challenge very well because he's young. He's only 62 years old, and he's already had 109 injections. We can imagine how many injections this patient will require because he was getting injections every 4 to 5 weeks, and there was still some fluid despite those. On 022, that fluid began to resolve and was fully resolved by week 12. This patient received no rescue injections, and the retina remained completely dry through week 24. This picture was really commonly observed through the first cohort of 6 patients. 5 out of the 6 patients had a complete response, meaning fully dry retinas from a single injection of 022 through 24 weeks. Overall, 0 rescue injections required. ADVM-022 was safe and well tolerated. The mean vision was stable, the mean CST was reduced. Based on these exciting preliminary data, we believe that 022 offers transformative potential to greatly reduce treatment burden and improve retinal anatomy with a single intravitreal injection in wet AMD. Thank you very much.